Good afternoon, my micro class. Now we're in chapter 5. This is the first chapter of our second series. So you're taking an exam over chapters 1, 3, and 4, and now we're going into chapter 5. And this one is all about how to keep microorganisms alive. So what nutrients, what growth factors they might need to survive, and then how they grow in general. So in the external environment as well as in a lab. So I always like to start by showing this cool picture that shows the bacterial cell cycle. In the eukaryotic cell cycle, we have growth phase 1, synthesis, growth phase 2, and then mitosis. But in bacterial cell cycle, we have period B, where the cell increases in mass and size, so very similar to our growth phases. Period C, where DNA replicates. So instead of calling it the S phase for synthesis, they call it chromosomal replication because bacteria tend to only have one single chromosome. And then we have period D, synthesis of the septum to form two identical cells. So microbial replication can be very rapid compared to eukaryotic replication. Microbes can microbes do double in number, and they typically double exponentially. So one cell becomes 2 cells, 2 cells becomes 4 cells, 4 cells becomes 8 cells, 8 cells becomes 16, 32, 64, 128, and you can just go on and on and on. And this can happen very rapidly. So bacteria can replicate in a matter of minutes a lot of times. Not all bacteria. Some bacteria are very slow at replicating, but some are very rapid at replicating. Example, E. coli replicates every 20 minutes, whereas some of your more pathogenic species take a little bit longer to replicate. So that's kind of a good thing, I guess. It gives us the potential or the chance to survive when there is an infection. So I like to give you a few mathematical equations. I like to give you a mathematical equation. I like to give you a few questions where you have to use that math equation. I know that not everybody loves math, but it's not as hard as it looks. So the reason I do this is because 10 cells can double, or not double, I won't say double, can become 40,000 cells in just a period of a few hours. And how many of you tend to, at a picnic, put all of your food on ice? Most people don't. I don't. If you're eating, if you're doing picnic food and you got, hot dogs, hamburgers, potato salad, you might put them in a room where the flies don't get at them, but you tend to just leave them sit out on the counter, and then people come in and grab what they want when they want to. And that's normal for us. That's what we do. But it can be potentially dangerous because bacteria can grow on food. And they can grow on food and double in numbers. And that can cause problems. So 10 cells can become 40,000 cells in a period of just a few hours. So how does that work? This is the equation that we use to calculate growth of bacteria, or generation time of bacteria. So we start with NT. N sub T means the number of cells in a population at any given time T. N sub 0 is the original number of population of cells in a population. So this would be before any replication occurred. 2 to the nth power is the number of divisions that have occurred. So we used pathogens at a potato or in salad at picnic for 4 hours, and we started with 10 cells. So, and let's assume that there's a 20-minute generation time. So that means that they double every 20 minutes. So n sub 0 is 10. That's this number. The tiny n is 12. And we know this because for every 60 minutes, we have 3 divisions because it's 20 minutes. So 20 times 3 is 60. And there's 4 hours. So 4 times 3 is 12. I will do this on the board for you. Just ask me to, because this gets confusing and people do struggle with this. I get it. So now we have n sub t equals n sub 0 times 2 to the nth, or 10 times 2 to the 12th. So if we do 2 to the 12th power times 10, we get nt equals 40,960 cells. So now I'll let you figure this out on your own, okay? And then when we get together, you can ask me about this. When we study microbes, we tend to look at microbes in a laboratory. It's easy. We can grow them, and because we are setting all of the conditions, they grow really well for us. But in the external environment in nature, microbes don't grow the same. They can't. They don't get everything they need. Humans aren't out there making sure that there's enough of every type of amino acid that the microbes need, and every micro element that they need. Because of this, they grow typically in groups or colonies, like biofilms, which we've already talked about. They can live as single organisms, but most live in communities or biofilms. And biofilms are what produce the slime on the sink tub, scum in a toilet bowl, stuff like that. On your teeth, it produces plaque. And biofilms are problematic because when we try to kill off pathogens and they're living in a biofilm, if that biofilm is in our system, the antibiotics don't tend to kill many cells. They might get a few of the very most superficial cells, but the majority of the cells they don't get. And because of that, the cells can live, they can replicate, and they can keep releasing whatever toxins that are causing our symptoms. And so I do want to mention, and I say this a lot, but bacteria aren't evil. They don't get into our system with the expectation that they're trying to kill us. Bacteria are just living. They'd be considered neutral, if anything. So you have, good, neutral, and evil. They would be neutral. They're not trying to hurt us. They don't care if we're alive or dead. They just want to survive themselves. And so they'll do whatever they have to do to survive. Make sense? And if not, ask me about it. All right, here's a video we'll play.
Clean abiotic surfaces immersed in liquid tend to attract and concentrate nutrients, whereas biotic surfaces such as plant or animal tissue can themselves be a source of nutrients for microorganisms. A biofilm is formed when microorganisms attach to and grow on a variety of biotic and abiotic surfaces. The initial attachment is usually reversible and often involves attachment by flagella, fimbriae, or other cell surface structures. This is followed by growth of the microorganism and production of an exopolymer, which makes the attachment irreversible. Growth within the biofilm is not uniform. Channels develop which permit incorporation of fresh nutrients and oxygen. Metabolic activities of microorganisms in a biofilm can create changes in the microenvironment. For example, an oxygen gradient can develop that permits anaerobic metabolism near the interior and aerobic metabolism on the exterior. Nutrient and pH gradients are also produced. In the natural environment, biofilms often consist of different types of organisms that function together in the cycling of the elements. For example, breakdown of cellulose on the surface of a decaying plant may result in release of glucose and other carbohydrates, which supports the growth of non-cellulose degraders. Carbohydrates are converted to organic acids and methane by anaerobic bacteria, and the methane can then be degraded by aerobic microorganisms.
Hello. There we go. Okay. So let's talk about how microorganisms grow in our lab, because that's really what we typically study. In our lab, microbes grow. They have a specific growth, I guess, what do I want to call it? Growth progression. That's written on the top of your slide, so that sounds good. And there are 4 phases to this growth progression. The first phase is called the lag phase. So this is the time that that it takes bacteria to get used to their new environment. So think about when you took bacteria, when you were doing your aseptic technique, and you took bacteria from one medium and you put it into another medium. They don't replicate immediately. They're going to get used to the new environment, and microbes have to get used to new environments all the time. They have to be really adaptable to survive. Once they finish getting used to their environment, or once they get through that lag phase, then they start replicating rapidly. And this is called the log phase, or the exponential growth phase. So the cells are going to replicate at whatever their growth rate is. It will be exponentially. This is typically when bacteria are most susceptible to antibiotics. This is also when diseases start to be seen. Bacteria will grow very rapidly until they hit a stationary phase. Stationary phase is when the bacteria are using all of their nutrients, and so we're still replicating, but some cells are dying off at the same time. So the amount of cell replication equals the amount of cell death, and so we kind of plateau. You can't keep growing exponentially because there's just not enough nutrients in the environment. And then we hit the phase of decline. So decline is also an exponential phase, but it's a much slower decline than you have for a log phase. So you slowly die off. And this is, typically it's in that stationary phase that we're transferring our bacteria to a new medium so that the cells can start growing really well again. There are a lot of different factors that we have to take into consideration when we're growing microorganisms. Again, in the environment, bacteria don't always live in the most the most comfortable or the most beneficial habitat, because the environment doesn't give bacteria what they need. Bacteria have to survive in the environment even though the conditions may not be beneficial. In our lab, it's a lot different. We can give them exactly what they need. So we're going to look at 4 basic variables that influence growth rates of microorganisms. Their temperature. Remember, we use an incubator that allows microbes to grow most effectively. The atmosphere, the amount of oxygen in the environment. The pH, so how acidic or alkaline the environment is, and the amount of water, so osmotic pressure, how much water or salt is in the environment. Organisms that can survive in very extreme environments are termed extremophiles, and these are typically your archaeans. Example of extremophiles would be hyperthermophiles, organisms that grow at really hot temperatures, or acidophiles that grow at very low pHs. So here is a figure from your textbook that looks at the temperature requirements of different organisms. So here we have psychrophiles. Psychrophiles are organisms that grow best at about 10 degrees Celsius. So it's pretty cold, but they can grow at 0 degrees. They can grow all the way up to about room temperature, about close to 20 degrees Celsius. Then we have psychrotrophs. Psychrotrophs grow best at about 20 degrees Celsius, but they can grow up to 30 degrees Celsius, so close to that room temperature, or close to body temperature, not quite. And they can grow in the very cold environments as well. Mesophiles are organisms that grow best at about 35 to 37 degrees Celsius. They can grow all the way up from about 10 degrees Celsius to close to 47 degrees Celsius. And then we have our thermophiles. Thermophiles are organisms that can grow, again, best at about 63 degrees Celsius, but they can grow anywhere from close to 40 all the way up to almost 75 degrees Celsius. And then the hyperthermophiles. Hyperthermophiles are organisms that grow best at around 100, between 90 and 100 degrees Celsius. They can grow at slightly higher and down to around 70 degrees Celsius. So large range. If you look up here, you can see archaea span the entire group. So you have archaeans that are psychrophiles all the way to hyperthermophiles. You have bacteria that can be psychrophiles all the way up through thermophiles, and some hyperthermophiles, but not much. And then we have eukaryotes that can be psychrophiles all the way through thermophiles. We follow under the eukarya, and our body temperature is about 37 degrees, so we would be considered the mesophiles. Psychrophiles, I just said this, but psychrophiles grow optimally, at a lower temperature. And these are the most common prokaryotic organisms, not the most common problematic organisms, though. Mesophiles are the organisms that grow typically at best at body temperature. And so these are the ones that live in the human body and can cause disease. Then we have thermophiles and hyperthermophiles. Something I do like to mention, the organisms in the psychrophiles, these are the organisms, between these and the psychrotrophs, these are the organisms that cause the majority of our spoilage in refrigerators. So I guess I'd say psychrotrophs tend to be the more spoilage type of organisms. but psychrophiles and psychotrophs can both spoil food very effectively. Microbes have varying needs for the environmental gases. Some organisms need a lot of oxygen to grow. Some cannot grow in the presence of oxygen. And so if there's oxygen in the environment, which at least in where we live, there's always oxygen in our gas mixture. And what was it called? In the air. Holy cows, I'm sorry. That was like a total brain meltdown for a second. We always have oxygen in our air. We can grow well, but organisms that can't grow in the presence of air would either not be able to grow at all or could potentially die. And so I like to mention this because some people don't know, but oxygen is deadly. Oxygen is actually a very deadly gas, but we need oxygen to survive. So if we have oxygen in the environment, we're going to use it, but we have special enzymes that protect us from the oxidative activities of oxygen. But not all microbes have these special enzymes inside. And so I'm just going to go through the different types of organisms and what they need. So in this C here, this is an organism that grows with oxygen, cannot grow without oxygen. This is called an obligate aerobe. They need oxygen to survive. If there's no oxygen, they will not grow. This here under A is an organism that grows best with oxygen, but if there's no oxygen in the environment, they will still grow. This is a facultative anaerobe. So facultative anaerobes grow best with oxygen, but will grow without oxygen as well. So A and C are the two important categories that we see in our lab. Them are the only organisms we have. B is called a microaerophile or a capnophile. These are organisms that grow, they need oxygen, but at a very low percentage. So I think it's like 85% oxygen levels, where we need a lot more oxygen to survive. Or where, I shouldn't say we, obligate aerobes need more. And then over here we have obligate anaerobes under E. Obligate anaerobes grow without oxygen, but if there's oxygen in the environment, they will not grow at all. And then here is an aerotolerant anaerobe, organism that grows in the presence of oxygen, grows just as well as in the absence of oxygen. It doesn't use oxygen, but it has enzymes that can convert the oxidative properties to water that is not, or to other enzyme, I won't say enzymes, other molecules that won't damage them. I just talked about these. I talked about obligate anaerobes and aerotolerant anaerobes. We're going to use a special broth in our lab this semester called thioglycolate broth. And that broth is used to test oxygen sensitivity in different microorganisms. Oh, wow. I have a misspelling here. All right, I'm going to go to the next slide. I'm going to actually stop here and go and make a new video for the last part of this chapter, okay? Bye.